Hello, everyone. Welcome. So welcome or good morning, good afternoon, good evening to wherever you are in the world. I can see the numbers are still climbing up. It is 7 o'clock and typically we give it a little bit more time before we really jump into the topic. So whilst we wait for this first webinar, the first Sophion hosted webinar in 2023 to start, I'm just going to play a little video about some of the technology that's going to be featured in this webinar, just so we give some of the others a chance to join, and then I'll start the meeting officially.
84 is an automated patch clamp system designed for drug discovery when you need high throughput and high resolution. With its high fidelity recordings, accurate liquid handling, and unattended operation, you can combine high throughput screening with complex ion channel interrogation. And for basic research, the Cube's high throughput strengthens the statistical power of difficult assays. Intelligent compound management ensures that Cube always dilutes just in time. This mitigates the risk of compound adsorption to surfaces. 384 amplifiers provide individual and adaptive patch clamping conditions to ensure tighter data distribution and more predictive results. Q-chips have microfluidic channels for fast and complete liquid exchange. Each Q-chip contains 384 individual electrode pairs that never drift, never require maintenance, and are always ready for use. The Q-chip design allows an endless number of compound additions. This makes it very convenient to use for complex protocols and extremely cost-effective for users. Cube 384 offers true walk-away operation. You can test more than 6,000 wells with just one click. The Cube does not require the skills of an electrophysiologist to operate, and it can be learned in a single day. The Cube's patented automatic cell preparation unit enables longer runtimes from a single harvest of cells. While recording, the temperature can also be set at any desired level and kept constant. Series resistance compensation up to 100%, precise voltage clamp, current clamp, and stacked liquid addition are the basis for full-scale ion channel research and are only provided by Cube. The Sophion Analyzer software is designed to be intuitive and simple to use. Once you have set up your analysis, all subsequent runs are automatically analyzed without any manual intervention. And with filters for data quality control, you can manage large amounts of data very quickly. With Cube 384, your data is automatically backed up, and the size of the data can be reduced to save valuable space on your backup system. Despite many advanced processes and components included in the Cube system, everything is controlled from one computer. We design, develop, and manufacture our instruments in Copenhagen, Denmark. And a dedicated global team of application scientists and field service engineers are ready to help you whenever you need our support. If you are interested in learning more about Cube 384, please check our website, sophion.com, or send an email to info@sophion.com. You are also very welcome to visit our labs in Copenhagen, Denmark, Boston, USA, or Honjo, Waseda, Japan to see and try the Cube 384. Cube 384 is an automated patch.
All right, once again, welcome to this webinar. I see now that more people have joined in. So this is the first Sophion hosted webinar in 2023. And from the number of attendees that we've received here from the entire world, I can see that this is a topic of high interest. So how this is going to go down today is we have two presenters myself, I'm going to give a small introduction where I'm going to introduce some of the tools that you can be using for pain drug discovery. And then I'm going to hand it off to our main speaker of the session. And for this, I'm going to start actually sharing my presentation here. So for this, I'm going to hand it over to Dr. Reza Ghovanloo. So I'm going to introduce Reza already now, then he can later on overtake it seamlessly. So Reza has received his PhD from Simon Fraser University in the lab of Professor Peter Rubin, where he studied the biophysical and pharmacological properties of voltage-gated sodium channels. So during his graduate training, he also spent time at Xenon Pharmaceuticals and SciLife Lab. He has authored research and review articles describing different channelopathies and the effect of pharmacological agents, such as various cannabinoids on ion channels. He's now a CIHR Banting postdoctoral fellow at the Yale School of Medicine in the renowned laboratory of Professor Stephen Waxman, and he studies the molecular neuroscience of cells. Pain. Okay, don't know where my head is this morning, but Reza is going to present a fantastic work on a novel approach that allows him to record currents from isolated neurons. So a few housekeeping points before we start. You can please use the chat function to post your question, and then at the end of the session, we're going to be going through all the different questions that were posted and answer them then with both speakers on the board. You also have a handout function where you can download some of the features, some of the posters, for example, that Reza has recently presented at a Biophysical Society meeting, and other documents that might interest you. And without further ado, I'm now jumping into the presentation. So with a problem statement. So chronic pain is defined as pain that lasts for 3 months and does not remit as healing occurs. As you can see here in that left box here, pain affects more than 30% of Americans that have some form of chronic or severe pain. And if you take this together, that is actually more patients than cancer, heart disease, and diabetes combined, which is really a shocking number. So current treatment options are limited, and they come with several unwanted side effects, including a high addictive potential of the opioids that are often prescribed. And when you look to the right side, you see some recent statistics from the year 2019, where here it was found that more than 10 million people in the US misused prescription opioids in that year. And that then fed into the left number here, where more than 70,000 people unfortunately died from drug overdose. So I think all of this points towards the need for novel, non-addictive pain therapies. So let's look briefly at the pipeline, the drug developing pipeline for pain medications. And here a recent bioindustry analysis report that came out in February compared the venture funding for different indications and the prevalence of it in the United States. And looking at this top left graph, I think you can easily see that pain has a very unfavorable ratio here with a high prevalence, but relatively low venture funding, where cancer has a significant funding, but not so high of a prevalence. Of course, it's also a more devastating disease in parts. So this is also sort of reflected in the clinical pipeline, where we see here 2 bars representing the clinical programs for pain in 27 versus 2022. And I think you can appreciate that there was almost a 50% decline throughout all the steps of the clinical development process. And there are several reasons for that. So one of which is shown here in this success rate depiction, where we see that between 2013 and 2022, in average, all medicines that entered clinical evaluation 6.5% made it to the FDA approval, where in relation, pain medication has about a tenfold lower likelihood of succeeding. And there are, of course, a plethora of reasons for this challenging development, but one of which is that some of these assays, the solid science that is happening in the in vitro, does often not translate well over into patients. And for this, I think we really would like to contribute and share this webinar today to potentially provide a little bit of a different side of how this can be fostered or improved in the future with better assays. If we look at some of the targets that are being pursued by the industry, you see such an overview here. There are, of course, more than that, but these are some of the major ones. And we see that from 2017, there was a dramatic change from many companies pursuing opioid receptors to now many have dropped out in 2022, since that target class really has come out of favor. In contrast, sodium channels, and in particular voltage-gated sodium channels, they seem to receive still a high attention in the industry, with now receiving most of the programs are actually on sodium channels in pain medication development. There are other ion channels that, of course, are for our interest here: TRP channels, glutamate receptors, GABA receptors, and calcium channels, although calcium channels have recently also seen less attention due to some failures in the clinic. So why are voltage-gated sodium channels so attractive? And here I want to show, this is actually a figure from an article that came out of the Waxman lab that very nicely shows the contribution of various voltage-gated sodium channel subtypes to the action potential in DRG neurons. So action potentials are the real currency of pain. Action potentials, they propagate the pain signal to the brain, where it's then getting interpreted as pain. And so you can see that voltage-gated sodium channels, they play critical roles in initiating and propagating this action potential in different phases. And some of these ion channels, they really have exquisite human genetic validation for their relevance in pain. And in particular, the NAB 1.7, which the group of Dr. Waxman studies intensely. Several loss of function individuals were identified where the channel doesn't function as in a normal state, and these individuals experience sometimes no pain at all, where gain of function individuals were also identified, where these individuals suffer from severe or intense periods of pain. So this is a very attractive target class to go after. And the patch clamp technology really lends itself to investigate and develop drugs on this target class. And I want to use a little bit of time of what kind of tools are available to develop drugs here. So the patch clamp technology is so well suited for that, because it allows to precisely clamp the membrane potential. And for example, in this way, one can study different states of the ion channel. So when we look at this insert here at the lower left side, clamping a cell to this 2-pulse step protocol, we will first come from a closed state and activate all the channels at P1, which is then results in that large signal, that large inward current as shown in the larger plot here. When we then step back to a partial inactivated potential, where some of the channels enter the inactivated state, and we do a second depolarization step to stimulate the channels, we will see that only a subpopulations of channels are available to be activated, giving rise to this smaller signal, as we can see on the right side. So if there is pharmacology that preferentially binds to, let's say, the inactivated state, then this will show a more potent IC50 value the longer the channel spends in the inactivated state, or the more channels are in the inactivated state. So here on the right is an example, amitriptyline, that has exactly such a state-dependent effect, where we can see that when we go in this intermediate step to -65, where many of the channels are in the inactivated state, we see a left shift in this concentration response curve, which results in a significantly more potent IC50 value. So to study that, really, we at Sophion have developed a feature some time ago that really allows you to address this in a more precise way to get better results during your development. And we call this adaptive voltage protocols. These adaptive voltage protocols allow to interrogate a cell in the beginning of the experiment, determine a biophysical parameter, and then feed it back to that same cell later on in the experiment. So with more detail here, one can apply a family of voltage steps and then set an online cursor here at this last depolarization step. The current that's been read out here can then online be fitted with a Boltzmann curve here, and we can ask the algorithm to extract the V-half value, the value at which 50% of the channels are sitting in the inactivated state. This will allow us to get 48, or in the cube system, 384 individual V-half values for each cell. And this can then later in the experiment be fed back in that two-step protocol that I've introduced in the previous slide, where we step up from the closed state, but then we go to a empirically defined voltage value that's different for each cell, and then we do another stimulation. So what is the benefit really of this feature? This can be shown when we divide the 0.2 by 0.1, then we will get such a distribution as shown here when we first pay attention to that standard method. So traditionally, researchers, they would choose a literature value, choose one value where they hold the channels or the cells, or clamp the cells at the V-half value, but this is throughout the entire experiment. Every experiment receives the same value. With that, you have some biological variations. You also have some technical variations with regards to access resistance, and that gives rise to this large spread in this distribution here. On the opposite side, when using the adaptive feature where the value was empirically defined for each cell, we are really narrowing down this to the desired value and have this 50% availability. This then translates immediately into the pharmacology, as you can see here, for 6 different reference compounds that were tested on the cardiac sodium channel, but it could be done for the pain-related ones as well. And I hope you can appreciate that using the different approaches, not only do we get much tighter data, sometimes it even brings up a completely different value here. And that may be the difference between a hit or not a hit in your screening campaign. With that said, it is, of course, all the vast majority of drug discovery campaigns, they start out with a screen of many thousands or hundred thousands of compounds, and for this, automation is really critical. Traditionally, Flipper or other fluorescent-based technologies have been utilized to screen through a lot of compounds and triage them to identify some hits to further process them in the cascade. With the Cube that was released already some years ago now, we now have the possibility to really increase that throughput. And this is also done by the fact that we can do cumulative application of compounds. So on the left side, you see here a recording from the voltage-gated sodium channel subtype 1.7, and the peak currents are being plotted over time. And we have added increasing concentrations of tetracaine here as indicated on the top here, and you can appreciate the gradual inhibition of this current leading to a full diminishment of this current. So one reason why this is possible is because the technology features a flow channel architecture in the consumable, where we have an inlet, and then this solution is being guided by the cell that is sitting on this patch hole, and then into a waste well. And this allows us to add an indefinite amount of solution and completely remove it again during the experiment. So in the setup as I've shown it here, we were able to record up to 384 individual concentration response curves, which really allows you to test a lot of compounds in one go. Another challenge with developing drugs on voltage-gated sodium channels is the fact that a high occupancy is required for these channels. And this is because it actually requires a higher concentration or higher IC50 value to inhibit the action potential, the currency of pain, as it does to inhibit the sodium channel itself. So there's a reservoir, so to say, of sodium channels. And to really address this parameter, one way how researchers can about is to develop drugs that have a long residence time. That means that once they are binding tightly to the ion channel, it takes long time for that to come off again. And such a parameter can be assessed with the microfluidic flow channel architecture, when we here again showing voltage-gated sodium channel 1.7, and we do the peak current adding TTX, the tetrodotoxin, you can see that this inhibits the current, but then we can completely wash this out again. And this allows one to make a fit to this wash in and wash out period to define the on and off rate, and therefore define critical kinetic binding characteristics. So that has been done by a group in Vancouver, by Banker et al. That is Xenon Pharmaceuticals. They have used exactly that approach on the cube, but they've washed in different concentrations of their molecules to then define the, or to then calculate the K on and K off values, as you can see here on the left, on the middle and the right plot. And then they were able to identify compounds that have a particularly long residence time that then allowed them to gain an improved efficacy in their drugs. So this is really going away from some of the more traditional evaluation where one would only assess compounds by potency, but here we can go more into detail of this mode of action. So I mentioned that high throughput is a critical part, and it's of course nice to see that there are groups who utilize automated patch clamp in a primary screening setup. This study here that came out of from a group at Pfizer Nucentis already in 2016 utilized the Cube system for a 158,000 compound screen on the NAV 1.7, and this was now actually recently been beaten, that number, by a report that came out of Charles River Laboratories, where the group has screened 730,000 compounds on the Cube. That data or that experiment was just presented at the SLAS conference, although the target and the indication was not released at this point. But it just goes to say that the technology has really evolved over this past decade, that automated patch clamp can indeed be used to screen in a primary screening settings to potentially reduce the development cascade, because more relevant chemical matter can be identified early on. So whilst recombinant cells that I've just, all the data that I've shown that came from heterologous expression systems, they are very relevant in particular for primary screenings, but further down in the cascade of development, one would like to move to some more physiologically relevant assays. And one technology that really has evolved over the past 15 years is entering the stage was the induced pluripotent stem cell technology. That is a very attractive technology, as it allows to take cells from any donor, from patients exhibiting diseases, but also from healthy donors, reprogram these cells and then differentiate them again into any cell in the body, which makes them very attractive tools for drug development. But pairing them together with the automated patch clamp technology offers an attractive way to really test a lot of these cells in a short amount of time. And exactly such a project is what we have set out to do with in a collaboration with Anatomic Tech. That's a company here in the US, a premier provider of iPSC-derived nociceptors. And we had their real DRG neuron product in the lab. And my former colleague developed an assay studying these or electrophysiologically characterizing these cells. So what you see here is a voltage clamp recording where we first applied a family of voltage steps to evoke this characteristic transient inwards current that goes, which is produced by the voltage-gated sodium channels. And you can appreciate that in the blue trace, when it was exposed to tetrodotoxin, the large portion of this current was inhibited, which points to the fact that these are TTX-sensitive currents in these cells. One would, however, expect also TTX resistant currents in these DRG neurons, and we were quite happy to see that in a subpopulation of these cells, we were able to detect this current. As you can see here, the red trace being the baseline, then the blue one showing the TTX inhibited current. And when we then use this A803467 compound at 10 micromolar, we were able to further reduce this current. And you can see in the current voltage relationship on the right that this is also in the average plot being shown here. So I mentioned in the intro that really we are not so interested in inhibiting sodium currents, but instead the result, the aim has to be to inhibit the action potential. And with that, it's nice that you can use these iPSC-derived cells to also study action potentials in the current clamp mode. So that was done here as we applied a ramp, a current ramp to these cells. And you can then see that we've seen a repeated firing of these in these cells, which really carry the morphology as you would expect it, but then go into more positive or to higher current injections that leads to depolarization, and then the signal is diminished here. I do have to mention that not all cells had so many repeated firings. There were several ones that did already diminish at an earlier point, which points to somewhat of an immature phenotype of these cells. So we did characterize these cells then and quantify that at different time points in culture. So these cells were received cryopreserved from Anatomic Tech, and then we exposed them to some growth factors in our culture in Bedford, Massachusetts, and then lifted them in suspension in different time points, as you can see here. And we were actually quite happy to see that we obtained a solid success rate with about 50% successful recordings that were achieved. This dropped a little bit when we went to later time points, which is mainly due to the fact that the cells formed a more complicated network, which makes it more complicated to lift them in suspension and to get a pure preparation. So out of these successfully recorded cells, the majority exhibited voltage-gated potassium currents, but and 50% showed voltage-gated sodium channels and some action potential firings, which was quite nice. So whilst this is a very promising and fast developing technology, the iPSC-derived cells, and we feel that it certainly is at a stage now where it can be used to triage your compounds further, to test some of your lead compounds on. It does have still some shortcomings, as I mentioned, that some of the cells, they lack maturity. And because of this, I am very happy to hand it over to Reza, who is going to tell us about an assay using primary cells that potentially don't have these shortcomings here. So I'm stopping my presentation now. Going over here again. So exactly, Reza is coming on stage. Yeah, just a quick reminder before we continue here then, if you please use the chat here to post your questions, and then again a reminder handout section, you will find some information that you can download, or we also have a little pop-up that is showing up here on the, probably showing up on the left of your screen. But without any further ado, Reza, the stage is all yours.
Awesome. Hi, everyone. First, I'd like to start off by thanking Sophion and Daniel specifically for organizing this webinar and inviting me to give this presentation today. So I'd like to start off by a very simple concept, which is essentially the concept of what makes us living organisms and humans. You can make an argument that one of the most amazing features about our biology is that we can perceive the world around us. And from essentially listening to a relaxing piece of music to looking at a beautiful painting, we have to be able to perceive the world around us. And at the core of this is a biological process known as electrical signaling. And what's critical, one of the most critical features or components of this important biological process is a transient all or none process known as the action potential, in which signals get transmitted over excitable tissues. And today, of course, after decades of research, we know so much about this important process. For instance, the phases that it has, from the depolarization phase to the repolarization phase, and so on and so forth. And we even have learned about how or what proteins are involved in various components of the action potential. So the reason why we have been able to learn so much about this, about the electrical signaling system and more fundamental units of this system, has been thanks in large part to the gold standard technique, which is the patch clamp technique. And this technique has taught us invaluable information over decades in terms of how single ion channels, single proteins, in the voltage clamp mode work, how they function, when expressed, overexpressed in heterologous systems, and of course how the combination or the ensembles of these channels work together in concert to produce beautiful patterns of excitability. And at the core of it all is Ohm's law, which is V equals IR, in which essentially you can clamp down or up any given one, any one of these variables. If you clamp the V part of it, you get voltage clamp, which typically we use in heterologous systems to understand how a single protein of interest functions. If you clamp the I, you get current clamp, and you can look at beautiful action potential morphologies. And of course, R, or 1 over G, gives you conductance clamp or dynamic clamp, which we're not going to really talk about a whole lot. So I think the best way to look at this is the analogy of an orchestra, in which if you actually look at the current clamp part, it's analogous to the orchestra itself, where you have a bevy of various types of instruments working together, producing a beautiful piece of music. Whereas in the voltage clamp mode, you can think of it in terms of just focusing in on a single instrument, in this case, a violin. So how does this technique work? It's quite simple. You have essentially you in a traditional sense, you patch onto a cell. You have an electrode on the internal side, one on the external side, and you can essentially manipulate the environment, the electrical environment, to your liking. And the traditional setup obviously looks like this. And then we know this is a very low throughput and laborious process. So obviously the limitations that are associated with this technique, which you can make an argument for some types of studies, dare I say, make it somewhat outdated, is that it's obviously a very, very laborious and slow process. And obviously those of us who have experience with this technique especially when you're dealing with cells that are a little bit more challenging to work with. It can take minutes, 10 minutes, 15 minutes, sometimes even longer, to get a successful recording, successful quality recording from a single cell. Of course, by nature, when you have to pick a cell, it has experimental bias because you're selecting one cell and not all the other cells that look wrong. And of course, there are issues with the consistency in terms of the experimental conditions, because it's a human in charge of controlling everything, from the amount of exposures you let on with perfusion systems, and the length of time you wait until you record a particular protocol. All of these things are rarely identical, I should say, from cell to cell. And of course, you're not doing it simultaneously in a traditional sense. You're not looking at cells on a head-to-head comparison. It's very challenging to do. The other part of it is the transition from voltage clamp to current clamp is often challenging. It puts a lot of stress on cells, and it's hard to have high success rates with that. And of course, in many cases, if you're recording from primary cells that come from animals, you culture them on cover slips overnight, over prolonged periods, until they're ready to be patched. And of course, these cells are not really used to being cultured on a glass cover slip. They're used to being, in an intact animal. And so the longer you let them sit on a glass cover slip in an incubator, presumably, or I think in theory, you can make an argument that the molecular biology of these cells are going to change over time. And so our objective was very simple. Using automated high throughput platforms, and in this case, we used the Sophion Cube, we sought to develop an approach, a method that solves many of these problems, with the goal of being able to understand how freshly isolated neurons essentially electrophysiologically work. And the first application that we looked at was pain, and specifically DRG neurons. So before going any further, I think it's always, Daniel already touched on this, but I think it's always important to keep reminding ourselves about the importance of pain, and how much of an immense public health problem it is worldwide. It is severely life-limiting for individuals who suffer from it, and unfortunately, it accompanies many other conditions, from chemotherapy-induced neuropathies to diabetes, among many others. And so it is obviously an important contributor to the opioid crisis that is rampaging much of the world, including right here in the United States. And there are not really that many effective alternatives for opioids. So then the question becomes, if pain is such an important problem and it's such a widespread problem, why has it not been solved yet? Well, it's because it's very complex. It's a long pathway. Every step of the pathway, from the site of injury, from where the nociception happens, all the way to when it gets to the brain and gets processed down as an unpleasant feeling that we call pain. It's very complex. Every step is very challenging. And of course, as scientists, as researchers, as fundamental biologists, we tend to focus in on individual components. So we break it down into smaller segments. But I think conceptually, it's important to remember that pain, much of its complexity is in the fact that it has two conceptual fundamental parts. The biological part, which is basically all of these processes, how every single one of these steps work, is what we study and what we're trying to solve. But there's also a psychological and emotional component, which is very subjective, and it's a person by person thing. And so all of these things together make it a huge problem. And of course, for the remainder of my presentation, I'll be focusing on the DRG. So why DRG neurons? Well, DRG neurons are for the purposes of my talk today, they are among the most complex electrical biological units. They are very important biological units, obviously, in the pathway of pain. And what makes them interesting for our purposes is that they contain various ion channel ensembles and at different stoichiometries, which include things like sodium channels, potassium channels, TRP channels, among many others. And every single one of these proteins and many others, of course, have important implications on pain. And specifically with respect to sodium channels, these cells host a lot of different sodium channels from that are biophysically quite distinctive and quite interesting, which include those that are sensitive to tetrodotoxin and those that are not. And so on sodium channels, we know that they are predominantly expressed in a tissue-specific manner. We have subtypes that are expressed in the heart and muscles, and we have subtypes that are predominantly found in the central nervous system. But the classic sodium channels found in the peripheral system are NaV1.6 through 9. However, today we of course know that in addition to these classic subtypes, there is also NaV1.1 and NaV1.3 that could also be expressed depending on which cells you're looking at, what conditions you're looking at. So there's a lot of different sodium channels expressed in there. And so that brings us to the topic of today, which was our project to try to figure out a way to remedy some of these technical problems. The workflow and our approach was quite simple. We wanted to essentially take cells from animals and essentially study them head-to-head simultaneously and in as unbiased of a way as possible. And to do this, we had to utilize 3 to 4 adult mice in this case. The reason why we had to pull cells from multiple animals is because inherently for many of these these types of instruments, you need a high cell count. You need lots and lots of cells, and it's hard to get cells from a single animal in sufficient quantities, especially when you do purification and the rest of it. And so upon removing the surgical the surgical removal of the DRG itself, the cells were isolated, purified, pulled together. They went on the Sophion cube in this case, and we were able to obtain high throughput voltage clamp and current clamp data. So, as with any other experiment, certainly electrophysiological experiment, at the core of a successful experiment is a successful cell culture. And the person who deserves the credit for this work is my colleague Peng Zhao, who was the person who led the charge and made this happen. So what we were able to do, which was quite challenging, was essentially to try to hit multiple targets. Our goal was to obviously, as I mentioned earlier, to have as many cells as possible because we need high cell count. So essentially, you need as many cells as possible when you look at them. But more importantly, we realized that you need high cell purity. So essentially, we wanted to make sure that we have very few of these types of things like glia, junk, debris, that kind of stuff, and more cells, more real neurons, because the debris can essentially reduce your success rate tremendously. Next, we wanted to make sure that the cells are healthy so that we can obtain successful patch clamp recordings from them. Essentially, what we were looking for was to make sure that we get cells that look round and beautiful like this as much as possible, and not have these kinds of cells that look like they're about to disintegrate or they're vacuolating. And for the purposes of this particular project, we also wanted to make sure that we maintain neurons of various sizes. And so we do not filter out one particular size, one particular diameter of cells throughout the purification steps. The reason being, it's a well-established fact in that dorsal root ganglion neurons have various types of biophysical properties and properties at that on a size-dependent manner, on a diameter manner. And so to do this, we obviously used cell mesh 70 micrometer cell mesh systems, and we did density gradient, be a 15% density gradient, and we were able to achieve this. And on the experimental front, we tried a whole lot of different stuff. Some of these things are obviously well known to those who use automated patch clamp systems. We've used 20 millimolar calcium at the beginning of experiments, because 20 millimolar calcium, like high calcium, helps with, I believe it forms calcium fluoride, especially when you use fluoride on the intracellular side. And it gives you better seal quality, which is important when you're dealing with these types of cells. And then we performed a whole bunch of extra regular saline, our standard saline washes. We resuspended obviously the cells in full DRG media, and we left the cells. We actually took the cells to the cube in DRG media. We played around with the suction pressure systems to get better seal formation, and also get better whole cell. But this thing actually is very important. So we obviously we have disclosed the suction protocol that we use in the paper that was published. However, I should mention that depending on what kind of cultures, what kind of cells you're dealing with, how your cells look like, you might have to play around with this. And of course, we tried various types of electrophysiological protocols to see what's possible. And then we finally learned that you can run from each prep, from each cell prep, you can run 3 to 4 cube runs and still obtain successful recordings, which was a remarkable feat for increased throughput. And so, as with many other types of studies, the first thing we looked at was the activating properties of these freshly isolated primary DRG neurons. And I should mention that the credit for much of this work, for the analysis of it, for the development of a Python code, which I will get into a little bit later, goes to my colleague and friend Sid Tyagi, who wrote the code, which was quite helpful. So with activation, as you would expect, as I mentioned, we have different types of sodium channels being present in DRG neurons. We have cells that express predominantly one type, like TTX sensitive currents, and those that express TTX resistant currents. And here's a very interesting current trace, where you can see that there seems to be, whatever sodium currents are present in this particular cell, they seem to be very fast. You can see this, like the fast component of the sodium current, whereas with this cell, you can clearly see these two beautiful distinctive current traces, one that is slower, one that is faster. And so, excuse me, here's the distribution of the capacitances of the cells that we were able to obtain in our cohort of cells. And of course, we use the capacitance as a proxy for cell diameter, because the higher the capacitance, the more membrane essentially you have, which means that you have a larger cell, bigger diameter cell. And with the GV curves, as you can see them here, we essentially realized that you can't really treat every single the GV curve from every single cell the same way. Meaning you cannot treat it with a single Boltzmann equation. there are some cells that inherently are going to have distinctive enough sodium channels or sodium currents that will have to be fit with a double Boltzmann, and some that probably fit better with a single Boltzmann. And so the code that my colleague developed, using that code, we were able to fit every single data point from every single cell with both a double Boltzmann and a single Boltzmann. And if the fit was better with a double Boltzmann, it would get binned into this figure over here. And if it fit better with a single Boltzmann, you can see it over here. And you can see the beautiful diversity of current densities associated with all of these curves, with all of these cells that are displayed here. Now, next, on a more interesting front, we looked at the inactivation properties of these cells. And for these, obviously, as you can see here, we were able to obtain a much larger cohort of cells that ranged from 10 picofarads all the way to about 70 picofarads, 75 picofarads, whatever number that is. And we applied the same type of, single versus double Boltzmann type of thing. And you can see these curves. You can appreciate how, you have certain cells that produce curves that clearly look better when they're fit with a single Boltzmann, and those that have these beautiful biphasic curves, which tends to be the majority of the cells, I should add. So based off the distribution of the capacitances, you can see here, we bend the cells up into 3 different sizes. So those that were between 10 to 15 picofarads, 15 to 40, and above 40 picofarads. And you can see that the majority of these curves kind of like have these, again, these biphasic features in the smallest category. And as you go up here, it seems like there's more of a mix. And so the cells that fit better with a double Boltzmann, you can see that the smallest cells, the first midpoint of the double Boltzmann, were the most hyperpolarized compared to the biggest, which is actually an expected finding, because it is suggested in the literature that as DRG cells get larger, they tend to express more TTX sensitive currents, specifically NaV1.6, whereas NaV1.7, which has the most hyperpolarized inactivation curve of all of the peripheral sodium channels, peripherally expressed sodium channels, they are predominant in the smallest cells. And yes, so this is quite an expected finding, and it's very interesting. Now, when we get to the single Boltzmann, the cells that fit better with a single Boltzmann, there were we interestingly found no cells in the smallest category that fit better with a single Boltzmann, which is quite interesting because it suggests that there is essentially no cells that are composed of one type of sodium channels, which is expected, but more importantly, from sodium channels that are biophysically very similar. Now, remember, these are neurons. So even when you have a cell that fits better with a single Boltzmann, it could probably is due to that neuron expressing multiple sodium channels with similar biophysical features. For instance, NaV1.1 and NaV1.6. And obviously, the final thing you can see here, the non-inactivating distribution, shows that basically the final portion of the final part of these curves quantified here. And you can see expectedly that in the largest cells, you have the least amount of non-inactivating sodium currents, which is to be expected given that this bin is composes the cells with the most TTX sensitive and NaV1.6 channels. The next thing we did was we looked at recovery from inactivation from 20 milliseconds and 500 milliseconds. 20 milliseconds is known as, it's a very fast time course. So this is recovery from fast inactivation, really. And 500 milliseconds is generally considered to be an intermediate amount of macroscopic inactivation, with certain populations of sodium channels having entered fast inactivated and certain, depending on the subtype, of course, and certain populations being in slow inactivated states. And what we found here was, again, with the when fit with a double exponential, you can see that with the slow tau of recovery, there was a statistically different, significant difference between all three bins, which is again to be expected. Whereas when we look at the 500 milliseconds, you're basically not being able to pick up on any differences, which is again not an unexpected finding, because each one of the sodium channels expressed in DRG neurons have different voltage dependent and kinetic properties. And if you're essentially eliciting 500 milliseconds using a 500 millisecond protocol, you're getting mixtures of fast and slow with any one of the sodium channels that are expressed there within that particular cell. And so it becomes really hard to separate them out. And the next thing we did was to perform a pharmacological experiment using just TTX 500 nanomolar, which is saturating levels of TTX known to block all of the TTX sensitive currents. Again, this is like on the x-axis, you're looking at the capacitance distribution and you're looking at TTX sensitivity, and you can see that when you essentially get to the largest cells, where again 1/6 is expected to be predominantly expressed, you're getting much more TTX inhibition, TTX induced inhibition. However, the thing that is very interesting about this, I think, is that there is quite a bit of diversity, variability in every single one of the bins, which is consistent with what you would expect in a biological system. You never, or you rarely, I should say, can get like a very simple, linear type of relationship. And then finally, of course, we wanted to perform obviously a voltage clamp and current clamp within the same cell, and specifically looking at the action potential aspect of these cells. And so for this, we use potassium fluoride on our system. You need fluoride to be able to obtain successful seals. And expectedly, you can see that now the potassium currents are no longer blocked in the absence of cesium. And you see these beautiful outward potassium currents, which are there, which translate over to these, these probability curves going below 0. And then here obviously are the protocols. And within these cells, we used, after performing this IV protocol, we performed a ramp current injection protocol in which we ramped up current injection to elicit action potentials. And here are 3 sample traces of 3 different cells, the voltage clamp and the current clamp within the same cells. 3 examples of cells with low to high excitability. And we again performed the same type of double Boltzmann versus single Boltzmann type of analysis. And you can see basically the obviously the double Boltzmann distributions, single Boltzmann distributions, and of course with current clamp, the number of spikes as a function of capacitance. Again, there's a lot of variability here. So to compare our new method to the traditional way of going about this using manual patch clamp technique, I think there are certain things that are important to keep in mind. The throughput is vastly different. So essentially, if you perform, let's say, 2 preps in one day, which can take time, you can patch from up to about 200 cells in one day. Whereas in manual patching, typically it's about at best 10, 15, whatever cells that you can record from in a day. Perfusion, of course, is possible in both setups, which is quite important. However, again, like I mentioned earlier, here we're perfusing, every cell is being exposed to the exact same type of conditions and the same length of perfusion, which is hard to achieve with manual patching. It's easier to get voltage clamp to current clamp transition here just because you have a higher throughput, so you have a higher success here than you would be able to achieve with manual patching. Cell selection is with manual patching, there's obviously experimental bias. Here, the machine is blinded. It patches whatever cells it happens to patch. The data analysis, everything we do here is completely unbiased. So from the quality control filters that we use in the analyzer software of the Cube system to the open source Python code that we developed, everything is automated and minimally biased in any shape or form. However, I should mention it's important to always have somebody who is an experienced electrophysiologist, of course, to look at everything once the analysis is done. And here, obviously, you have subconscious biases of experimenters. You do not need to do overnight incubation here in with our method, and with manual patching, typically people do. And of course, head-to-head comparison, you really can't do it. It's very challenging to do manually. And here, obviously, everything, you're essentially patching diverse cells simultaneously under the exact same conditions immediately after isolation. So the data that I shared with you today came from 7 preps. We used 5 preps for voltage clamp experiments, 2 preps for voltage clamp and current clamp data that I showed. We were able to record from 80. So essentially per cube run, you get about 25 to 45 cells on each chip in our experience. So you can record from up to about 100 cells per day or per prep. And this is another thing that was very interesting. So sodium channels are quite fast, and especially when you have larger currents, the clamping speed on the cube is not capable of handling the activation as well as it does with some of the other protocols. So, which is why you're seeing these discrepancy in the numbers. But I think given that you're still getting a high number of cells, that is not a major issue. And you can obviously see the rest of the numbers. The number of preps, as I mentioned, is 2 to 4 per prep. And you can basically have the cells sit on the cube as you're doing run after run, and they last for up to about 4 hours. And depending on obviously what kind of protocols you're running, the cells do, the ones that last to the very end, they last for about 40 to 50 minutes, which is absolutely astonishing, I think. And so in conclusion, hopefully I've convinced you that we were able to develop a method that is higher throughput than the existing method, the traditional way of going about it, to investigate freshly isolated neurons. The method is fast, unbiased, and enables head-to-head analysis of diverse neurons. It enables you to get away without having to deal with overnight culturing. And of course, you can do voltage and current clamp within the same neuron, which is a very important feature. And with that, I want to give a very special thank you to my mentors, Dr. Steve Axman and Suleiman Depaj, who've been great mentors and great supporters throughout all of this, and my co-authors, colleagues, who deserve a boatload of credit for much of the success of this project, from Sid to Pong to Mark and to Emre and the rest of our center. And with that, I'd be happy to take any questions.
Reza, for this fantastic presentation. I definitely enjoyed it.
Thank you.
So we do have a number of questions here in the chat that I would just go over now. And so starting here from the bottom, the first one that came in was from Jim. To the rest here, if you do have some questions, please, feel free to type away and add them to the chat. We're going to stay on a bit of time to answer these. But the first one came in from Jim. What was the resting membrane potential with the iPSC-derived cells? So I can answer that question. We did see quite heterogeneous distribution for this. But they were all ranging sort of in the 40 to even up to 60 millivolt, and then you've seen a clear correlation to the firing potential that the more depolarized cells, of course, had much more channels sitting in the inactivated states and weren't so readily firing action potentials. So the second question came in from Silke, and Reza, that is for you. Is it possible to share the dissociation protocol to get pure DRG neurons?
Excuse me. Absolutely. It's on the publication. And if you have any questions, feel free to email us.
Right, and this paper I think came out in Nature Methods that features most of the data that you've shown today.
This is in Cell Reports Methods.
Cell Reports Methods. Okay, yeah, I think you've shown it earlier. All right, next question here. Could you tell more about density gradient to have nice DRG cells? What density did you use?
15% BSA. Again, all of these details are indicated on the paper, on the published paper.
And really, I mean, I have to applaud you on this process, because I mean the micrographs that you've shown there from before and after, I mean, this was just so striking, and I think that really sort of speaks to the relevance of this clear purification. This one piece, and I think I've asked you during the biophysics as well, is that if it really was true that you only used 75,000 cells per milliliter, which is significantly below what we would typically use with other cells. But I think, as you rightly pointed out, the secret was probably the pure preparation that you had. Or
exactly. So it's actually, when we were developing this, I was just aiming, knowing what we knew about needing higher cell counts. Certainly, with in experience with hexels, where you use millions of cells for a typical run, we were like, let's just get like as many cells as possible. But when you do that, when you try to get as many cells as possible, you also get a lot of debris in there, and the success rate was significantly lower. You might get like 1 or 2 cells. But when we actually started to go after higher increased purity of cells, so basically making sure that you just get cells and nothing else, that's when the success rate started to go up.
Nice. That is really fantastic progress. All right, so we have several questions coming in from Yuha. So were you concerned about possibly altering the channel expression profile due to the cell preparation process, the loss axon and only recording from the soma? Maybe take that one first.
So that is a great question. Obviously, one of the limitations of this technique is that it's not really possible to patch processes at all. As I shown earlier on the slides, the cells all look round. It's whatever recording you get is from somatic. And it's so on the expression profile part of it, I think you can look at it from both different angles. Did we look at it specifically? The answer is no. We did think about it, but the two different perspectives that I think that are important to take into consideration is that, yes, as you're purifying cells, putting them through filters and all that, presumably you're inducing some inflammation, and probably there is some difference in cells essentially like could be experiencing various molecular, biological, environmental differences. But on the flip side, when you're culturing a cell on a cover slip, like I said, overnight, I think an argument could be made that that also changes expression patterns, expression profiles of the cells, because they're definitely not in an environment that they're used to. It's completely artificial, not even close to what they're used to. On the process, patching the processes, it is definitely a limitation. However, I will point out that patching processes is also no easy task using manual patching from experience. So, but yeah, I think this is important to keep in mind that this method that I shared today is not really it's not a replacement for in the entirety of manual traditional recording. There are, of course, still applications where there are things that you can do better with a manual patch clamp still, depending on what kind of experiments, what kind of questions you have in mind. But for the essentially for the aspect of wanting to define populations of neurons, looking at head-to-head comparisons, like the kind of work that I described earlier, for that, I think this method is definitely the way to go.
Yeah, I think that's a very good point. What's your take, that I can maybe squeeze an own question in here? What's your take then with regards to drug discovery?
Oh, I think I think that's a very, very cool question. It's a very important question. That is actually what I'm personally most excited about in terms of future applications of this type of method. Obviously, I mean, as I showed with the TTX data, imagine if you exposed one particular concentration of TTX or any given compound to, say, like a heterologous, like just a stable 1,7 line or whatever line. You would on average get basically the same the same inhibition, right, on all of the cells. Here you saw those, the basically the trends going up and down. It's because these are neurons. They're kind of like, in a way, just how every person is different, they look different. Every neuron seems to be different, because they're not going to respond to the same pharmacological agent the same way. And I think that's a very important thing. And in fact, I think that is potentially one of the shortcomings of the way drug discovery has been done. We look at cells in isolation, but we don't actually look at cells within a particular cell environment head to head together. And so if you have a cell that is, let's say, selective for, I don't know, let's say 1,7, and you're looking at essentially all of these different diameters of DRG neurons, and you're going to have like certain diameters that express a larger amount of 1,7 versus other sodium channels, or maybe don't even express 1,7. You're going to see differential amounts of inhibition. And remember, each one of these cells has a particular role in the animal where it comes from. Maybe that cell was in charge of sensing one type of pain and not another type of pain. Maybe it was in charge of sensing something acute. And so essentially your drug is going to affect that particular cell with that particular functionality a particular way. So I think all of those things are things that could be developed further for drug discovery applications, but I'm quite excited about it.
So am I. No, that's a great considerations. So if we continue here with Juha had some more questions regarding the calcium. You mentioned that you used a high external calcium at some point, 20 millimolar. And he asked, was that essential? Did you also try it without it?
The high calcium? We did try it without it, but yeah, I think very early on, honestly, I switched over to high calcium, and so the handful of times I tried no calcium, I was not getting that much success relatively. And then honestly, I stopped not using it. And you know how superstitious electrophysiologists tend to get. When we find something that works, we stick by it. And high calcium is something that obviously, like people who use automated patch clamp systems, certainly I know the Cube users, they use it all the time. And in order to make sure that there is no residual high calcium left over, I did 3 washes after after the calcium perfusion on the Cube, which is sort of like typical to make sure that there's no residual left. So yeah.
So maybe I can also just add, I mean, for the recombinant systems, we have evaluated it several times, and we don't necessarily see the benefit with recombinant cells, but I have heard that with these more challenging cells, I mean, it just fosters that crystal formation with the fluoride on the internal side and the calcium on the external side. Exactly. But to us, it's for many applications on a Cube, we don't find that it's necessary. Although with these more challenging cells, I think I've heard before that there might be a benefit. So Juha continues with a question here. What's the benefit of using automated patch clamp compared to high density multi-electrode array in context of action potentials?
I think that's another great question. I think the utility is completely different. We use MEA in our lab regularly for various types of projects, experiments. They have completely different utility. I think obviously MEA has the advantage of you not essentially dilating out all of the intracellular side as you do in whole cell patch clamp. So the internal environment remains intact, which is very important. You can get obviously like reliable, firing properties in terms of a spontaneous activity of cells, or maybe even using things like light stimulation and that kind of stuff. But you do not get the in-depth, action potential recordings, actual recordings that you would get with the patch clamp technique. So the kind of information you get from either or is different. They can complement each other, of course, fairly nicely, but I don't think one is a replacement for the other in many contexts.
And I think it also sort of like adding to the point that you pointed out earlier with the drug discovery, where you can basically use or characterize individual cell types and put them together and then assess the effect on, characterized cells. I think that is really a benefit for the patch clamp technique, where you can sort of like identify cells that have a functional expression of a particular subtype, and then pull these together and see what's the action potential. I think that would not be accessible with a MEA platform.
Exactly.
So next question comes from Surajit asking about, Reza, I was wondering how fluoride or chloride helps obtaining better patches. I think in some way we answered it already with the high calcium, but I don't know if you want to expand on this a bit further.
No, I think we pretty much answered it. So with fluoride, fluoride is something that us in the sodium channel field, we all use it. People who work with other channels, many of them haven't even heard of it. Certainly certain certain potassium channels, actually you can't even use fluoride because it kind of like it depletes PIP2, and it kind of causes rundown of this potassium current where you can't use it. But for us in sodium channel, fortunately in the sodium channel field, we use it all the time. It's not very physiological, of course, and ideally I would like to not use fluoride if possible, but for the kind of applications that we're interested in, it doesn't really alter our channels of interest. And so the reason why we use it is because it helps with sealing, with like calcium, forms crystals and that kind of stuff.
Yeah, and I mean, maybe I can just sort of like piggyback on this a little bit. That is one reason where the Q-patch technology really has sort of like some strong capabilities, because it allows to form these giga-omic seal resistances without the use of fluoride. And so for especially like recording cardiac action potentials, we found that there the influence of the fluoride is more critical. We were actually not able to pick up right action potential morphologies with the use of fluoride, but this does not seem to be an issue also in our hands with neurons so much. But so for cardiac action potential recordings, we resort to the QPatch platform and then use physiological solutions. But I think, Reza, you're absolutely right. It really, you need to know the limitations and then use it for the application where it makes sense.
Exactly.
I'm continuing here. Michael has a question. I think I can probably answer this. Would it be possible to combine this technique with single cell RNA sequencing? And this is again, so we haven't any data available as of right now, but there's of course a push in the field and interest in the field because patch-seq would be very, very attractive on these systems. With the Cube system, that would not be possible, very likely, because some of the internal channels are combined together. Potentially, one could develop an approach extracting the material from the external side, but I would rate this as probably more unlikely to be successful. With the QPatch technology, we have individual internal channels. So here it is conceivable that one might be able to suck through the cell, the components at the end of the experiment, and then collect the contents of the internal channel and send it off for RNA-seq. It is hypothetical at this point. We have not looked too much into it, so I cannot give you a thumb up and say that this will definitely be working, but in theory, it might be possible. Definitely interesting. So here from Kimberly, there comes another question for you, Reza. Did you try patching DRGs from injured mice, rats with this technique?
From, sorry?
That was Kimberly.
Did I try patching from? Oh, no, we did not. We did not do injured mice or rats, but it's definitely possible to do it, I'm sure. That is, in fact, one of the things that I think we might have mentioned it in our discussion section as potential applications for this. So you can essentially do whatever you want to, the cool aspect of it is you can treat an animal, you can behavior in an animal essentially, either treated with, adeno-associated viruses or, treat the animal with drugs. And I guess in this case, injure the animal, and I guess it depends on the type of injury. I guess the former applications are definitely something that is possible if you're administering a drug or a virus or something like that. But in terms of if you're thinking about like a physical, like injure one particular nerve, and wanting to look at basically, how that would translate over. Remember, we need high cell counts, and we're pulling cells from animals. So you're going to have to be able to manage that part of it. So I think that will be the challenge. Theoretically, I guess it will be possible, but it will probably be challenging. It depends on the kind of injury you're referring to.
All right, very good. I have to applaud the audience here. There are questions pouring in, so I hope, Reza, you have just a little bit more time. I know we are already on time here. So one question that came in from Damon. Many thanks for the interesting webinar. I work with KV7 channels and I'm only just getting into sodium channels now. So apologies for what might be a stupid question. I'm sure it's not. In terms of drug candidates for targeting NAV channels, which feature would you say is more attractive? Subtype specificity, example, discriminating between 1.7 and 1.9, or state specificity, greater affinity, efficacy for inactivated channels? Or should you aim for a drug candidate with both?
Oh, that is such a wonderful question. And I think in the pharmaceutical sector, certainly with for instance, like the compounds that Genentech was developing a few years ago, I think the answer by the pharmaceutical industry is subtype selectivity. Subtype selectivity, I think you should really think about it in terms of structural selectivity, because all sodium channels are very similar in their overall, structure. And with, for instance, these selective class of compounds that bind the fourth voltage sensing domain VSD4 binders, the reason why they have been excuse me, of such enormous interest by the pharmaceutical industry is because they select, that part of the channels are different enough between the 9 subtypes where you can hope to target one channel and not others. So that's sort of like the idea. And as Daniel mentioned earlier, there has been problems with occupancy with those classes of compounds in terms of their actual success and efficacy in the clinic. Now, state selectivity or state dependence, that has actually worked. It's many of the traditional compounds that are in play, that are used, your lidocaines of the world, your phenytones of the world, all of those things are completely structurally non-selective, pretty much. And the reason why they do work and they are in use within in clinical settings is because they're administered properly, depending on which particular drug you're looking at. And these things are state-dependent compounds. They bind in the pore and in the local anesthetic site on the intracellular side of the sodium channel pore, and they work. So to answer your question, if you have subtype selectivity, which by the virtue of binding to that voltage sensing domain 4, which is in charge of fast activation in sodium channels, if you can have that function, it's fantastic. It's subtype selective, and it is, just because of where it binds, it's also going to be very state selective, state dependent. But so far as we speak right now, the clinical success has come from these non-structurally selective compounds that are state dependent. So I guess if you're looking for the next biggest thing, go for the former, but the latter is the one that is actually working right now. So hopefully that answers your question.
I think that's a great answer, and I think we're all still rooting for Vertex Pharmaceutical, who has a candidate for NAV 1.8 and a clinical evaluation, which then would be more the selectivity, I guess. But I think there are other approaches also being taken, and I think obviously needless to say, you want to have the selectivity when it comes to the cardiac sodium channel. That one should definitely.
That's the big no-no. It's every time something hits anything in the heart, whether it be 1.5, NAV 1.5 or HERG or whatever, that's usually when it goes to die. So yeah.
Should I pronounce his name? I'm probably gonna embarrass myself. So the next question comes in from Wachich. Apologize if that's not correctly pronounced. Does one need the analysis in Python, or would the Sophion analysis be enough?
We use both. So in terms of the quality, so we used the Boltzmann and like the fit quality features in the analyzer software to filter out bad cells, and you are going to get a lot of poorly clamped or poor quality cells on the cube. Obviously, these are neurons. They're vulnerable cells. They're not. I mean, hex cells, you can throw them in the washing machine and they're still going to come out working fine. But these cells are quite sensitive. So we did use all the features that were available to us in Analyzer, and if Sophion adds more features, I'm sure we will be using them too. But the reason why we had to also, in addition to the features in Analyzer, resort to developing our own Python code was because it just wasn't sufficient to do everything in Analyzer. And for instance, like the single versus double Boltzmann thing, that just does not exist in Analyzer. And we also did additional mathematical filtration in the Python. So you need both. But the code is open source. It's available online, so you can use that.
I think that's a great answer. And I do want to add here that we are, of course, always having an open ear for these kind of recommendations. I think, Reza, you've provided us already in the past with some valuable input. And so it is an evolving software where we are continuously adding these new features for these more advanced assays. But I would say at this point, it has already evolved over 2 decades. And the software is capable in many regards, but of course, there's always more we can do. So that's it. That's good input for us. So then we have Likhit asks, Reza, can you share your contact mail?
Oh, of course. It's my name dot my last name at yale.edu. Reza.Ghovanloo at yale.edu.
Right, and otherwise, what's on the paper maybe as well?
I'm not the corresponding author, so mine is not. But yeah, you can. My email is on the Yale website. You can look that up. Yeah.
Otherwise, if someone reaches out to Sophion, we'd also be happy to forward that. So the last question, then it comes from Silke again. Did you also patch fresh neurons from other species, for example, rats, monkey on the cube?
We have done rats. We have not done monkeys. Are challenging to get. Yeah, it works. It works with rats too.
All right. Yeah. Okay, that is good to know. That's good. Did you see any size differences between these?
Yeah. So that's actually that is something that is actually, I think, a very important important point. Depending on what age animal you use, and those individuals who have worked with cells from various species know this. The sizes of the cells are going to be different. The age of the animal is going to make a difference. Whether you're using essentially like I don't know, like we use 7 to 9 weeks in this paper, but if you use younger, for instance, the size difference is going to be different.
All right, that makes sense. All right, I think we had quite a good session. Actually, I forgot to put up our screen that we want to show here at the end, so I'll do it now. So I want to thank once again Reza for this very nice presentation and this great development that you've done. I'm eager, to see what else comes out of the lab and what you're producing next. But again, thank you for giving that presentation here.
My pleasure.
I also want to thank my colleague Jane Lucas, who has really put everything in the background together here with that platform. She's not visible here, but she's done a great job putting all of that together. And then I want to just point you towards some of the events that we have coming up now. We have in the coming months a webinar where we're going to introduce one of the latest features that is rolling out now with the new software. Now, first for the Cube patch, but we will also see that for the Cube likely in the future, which is the adaptive current clamp. It's in a similar way as what I've explained for the adaptive voltage clamp, but this helps to overcome some of the challenges with regards to too depolarized membrane potentials or leaky cells, and you will still be able to get current clamp recordings from these then. And then I also, if you happen to be in Japan, we are continuing our ion channel modulation symposium series that started in Cambridge, UK, and bringing it now over to Japan as well. We had it last year in the US. And then we are continuing here in June also with the session, which must be probably the 7th or 8th edition. I'm not completely sure, at Cambridge University again. So please, if you want a spot there, I'd advise you that you're signing up quickly because those seats are taken very, very fast always. All right, with that, then thank you for your attention and your interest in that. If there are any further questions, please don't hesitate to reach out to us. And yeah, we're looking forward to staying connected. Thank you. See ya.